Today, we're going undercover. We're diving into the invisible world of pathogens. You know, the microscopic culprits behind some of the world's most serious diseases. We're going to be the detectives here, but our main tool isn't a fingerprint kit, it's a microscope. So let's start the investigation. So right off the bat, here's the biggest challenge for pretty much every public health lab on the planet. Your suspect is an organism that's, 1,000 times smaller than a grain of sand? So how do you identify an enemy you literally cannot see? Well, you do it with forensics. And in the world of microbiology, the #1 forensic tool is something called staining. It's a really cool technique where you use special dyes that make these invisible little organisms just pop into view, revealing who they are, their shapes, and all their secrets. All right, let's open our first case file. This one's all about the most important first step in any bacterial investigation. See, before you can know who your suspect is, you first need to figure out what gang they belong to. And this is how you do it, the Gram stain. You know, this thing was developed over 100 years ago, and it's still the very first test labs run on an unknown bacterium. It works by checking out the structure of the bacterial cell wall. And with that one test, it splits basically the entire bacterial kingdom into 2 huge groups. This first clue isn't just about narrowing down the suspect list, it often tells doctors which kind of antibiotic is going to work. I want you to think of this whole procedure as a kind of chemical interrogation. So first, all the suspects get hit with this deep purple dye. Then, a mordant, think of it like a glue, is added to lock that purple dye in. Now here's the key step. A decolorizer comes in and tries to wash all that color away. It's basically a stress test for their cell wall. And finally, a pink counterstain is added to see which of the bacteria failed the test and lost that original purple color. And boom, here's the result. The suspects have shown their true colors. The gram-positive bacteria, which have these thick, fortress-like walls, they hold onto that purple dye no problem. But the gram-negative bacteria with their thinner walls, they just can't take the pressure. They lose the purple and get tagged by that pink counterstain instead. And just like that, our massive list of suspects has been cut in half. Okay, on to our next case. Now, some criminals, they just have better defenses. They're way harder to find, harder to identify, and they don't respond to the usual techniques. This case involves one of the most notorious pathogens out there. Yeah, we're talking about Mycobacterium tuberculosis, the bacteria that causes TB. Its secret weapon is this waxy outer layer that acts like a fortress. It completely resists the dyes that we use in the Gram stain, so it's basically a ghost in the system. We're going to need a specialized tool to unmask this one. So to breach this fortress, we need a special tool. The Ziehl-Neelsen stain. This thing is basically a chemical assault. It uses intense heat to literally melt that waxy wall open, forcing a really powerful reddish-pink dye inside. And once that dye is in, the waxy wall cools down and traps it. It just refuses to let go, even when it gets blasted by a powerful acid wash. And just look at this result. After that acid wash, the background and any other bacteria are stained blue. But the TB bacteria, the acid-fast ones, they're unmistakable. They are just blazing through in this bright reddish pink because they held on to that original stain. The fortress has been breached and the killer is caught red-handed. All right, let's move to our final case file. This time, the crime scene isn't in the lungs. It's the human bloodstream. And the culprit? It isn't a bacterium at all. It's a parasite, the one responsible for malaria. So the investigation kicks off by collecting evidence on a simple glass slide. 2 types of samples are taken, a thick smear, which is basically a concentrated drop of blood, and a thin smear, which spreads the blood cells out into a single neat layer. Both of these are absolutely critical to solving the case. Think of it like this, right? The thick smear is your surveillance tool. Because it concentrates so much blood into one spot, it's the best way to answer one simple question. Are there any parasites here at all? Yes or no? The thin smear, on the other hand, that's for identification. It preserves the shape of the blood cells and the parasites inside them, which lets us figure out exactly which species of malaria we're dealing with. And figuring out the species is so important because not all malaria is the same. This one here is Plasmodium falciparum, and it's the deadliest species by far. Its microscopic fingerprint, which you can see on the thin smear, is totally unmistakable. You see those unique crescent-shaped cells? They look kind of like little bananas. Finding those is a critical clue. Now contrast that with this one, Plasmodium vivax. Notice how its later stages are round, not crescent-shaped. It also has this weird habit of enlarging and distorting the red blood cells it infects. Spotting these subtle little differences is really the core of the diagnosis. It lets a technician identify the exact culprit and get the patient on the right course of treatment. So we've seen all this amazing detective work in action, but let's zoom out for a second and see how this all connects. How does a little colored stain on a glass slide become a lifeline for patients, and sometimes for entire communities? So why does all this microscopic detective work matter so much? Well, because identification is everything. A simple gram stain result can point to the right antibiotic from day one. A positive TB test, that triggers immediate patient isolation to stop the disease from spreading. And identifying a specific malaria species helps public health officials predict and even control outbreaks. This stuff isn't academic. It's the absolute foundation of modern disease control. And all of that data from individual patients, it gets scaled up. Every single diagnosis feeds into these bigger public health metrics, like the annual parasite incidence, or API. This is what allows countries like India to literally map out high-risk areas and direct their resources, like mosquito control, to the exact places where they're needed most. I mean, the scale of this operation is just staggering. To keep that crucial API metric accurate and to control malaria, India alone screens roughly 100 million blood slides every single year. That's 100 million individual forensic investigations, all done one microscopic detective at a time. And as this source points out, even with all of our fancy modern technology, these fundamental tools, the stains, the slides, the microscopes, they are still the bedrock of disease surveillance. They continue to be the main tools we use to understand these diseases and to fight back. For over a century, these techniques have been our trusted allies, unmasking invisible killers one slide at a time. It really just leaves you with a final thought. As pathogens keep changing and evolving, what new kinds of investigative tools will the next generation of microscopic detectives need to keep us all safe?